Hello and welcome to GeneLab Chat. Today we'll be interviewing Dr. Sabanayagam from the Delaware Biotechnology Institute at the University of Delaware. Dr. Sabanayagam and colleagues conducted genetic studies using C. elegans under simulated microgravity conditions in order to determine genes involved with microgravity adaptation. They compared their findings to 4 open science datasets. In this interview, Dr. Sabanayagam describes this research and how the GeneLab data systems and the AWGs contributed to this reanalysis of the open science datasets. Thanks for joining us, Dr. Sabanayagam. Could you please let our audience know the most important findings of your study?
Having on GeneLab chats. Sure. Our lab uses a worm called C. elegans to study biochemical pathways altered in microgravity, which we simulate in the lab by planar rotation. When you watch videos of the animals during planar rotation, we see that the worms are in free fall, much like an orbiting space station. We exposed the worms to simulated microgravity for 4 days and determined their expressed genes by RNA sequencing. When we compared our findings with 4 spaceflight studies, we found overlapping gene expression levels, with data from the CRISE 4-day mission showing the largest overlap with our experiments. One particular finding that we discuss in our publication is that the insulin pathway is altered in simulated microgravity. This pathway is associated with aging and longevity and has been noted to change in spaceform C. elegans. We looked for interacting pathways and found that the sphingolipid signaling pathway is downregulated in simulated microgravity and is also associated with aging and longevity. Downregulation of this pathway would lead to a decrease in total sphingolipids, which we later verified using liquid chromatography and mass spectrometry. In this study, we also wanted to understand how long these genetic genes persist in the worms after returning to ground. Our results revealed that while some biological processes are only affected during microgravity exposure, others can remain affected for at least 8 days after return to ground. For example, neuropeptide signaling pathway genes were expressed only in microgravity, whereas defense response genes remain downregulated for 8 days after return to ground conditions.
Thank you. And my second question for you is, how did the GeneLab data systems or the AWGs help assist you in your work so you could achieve these goals?
Since the space-borne data through GeneLab was absolutely necessary to validate our planar rotation experiment. As GeneLab contains raw data sets, we can push these through our own bioinformatics pipeline to reveal other features not possible in individual publication. For this study, it was interesting to see that our 4-day planar rotation experiment closely resembled the CRISE 4-day mission, more so than the CRISE 8-day or ICE-1st missions.
Thank you, Dr. Sabanayagam, for sharing your insights and that research with us. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access to 4 data sets analyzed in this work with the links provided in the description section below. Thank you and have a great day.